His Excellency, the Honourable Alex Chernoff, Governor of Victoria and Mrs Elizabeth Chernoff, the Right Honourable Malcolm Fraser, former Prime Minister of this country, and Mrs Tammy Fraser, Mr Carrillo Gantner, nephew of the late Kenneth Myer and Mrs Zian Gantner, Mrs Neilma Gantner, sister of the late Kenneth Myer, distinguished guests, ladies and gentlemen. Good evening. My name is Geoffrey Donnan. I'm Director of the Florey Neuroscience Institutes, and welcome to the 15th Kenneth B Myer Lecture. This is the second one we've held here at the Melbourne Convention and Exhibition Centre. The Kenneth B Myer Lecture is in honour of Kenneth Myer, who is the founding president of the Howard Florey Institute. The lecture was established in 1995, 3 years after his untimely death. The object of the lecture is to honour the memory of Kenneth Myer, but as well as that to bring to the Australian public science in a lay form. This is an important role, we think, for an institute such as ours in a modern, sophisticated and what we think is a clever society. The lecture today is to be delivered by Professor David Attwell, as you've seen in the advertisement. David trained as a physicist in Oxford before switching to study physiology and nerve and muscle, which is a common path, I must say. David then worked on the retina in Berkeley, California, before returning to the UK, where he became an academic at the University College London, where he became an academic becoming the Jodrell Professor of Physiology. He's a Fellow of the Academy of Medical Sciences and also of the Royal Society. David's well known for his work on brain energy use and how the brain uses electrical signals to think, how you and I think, and providing a better understanding of the cellular events leading to nerve damage in conditions such as stroke. So it's very appropriate tonight that the title of his lecture is indeed Brain Power. So without further ado, ladies and gentlemen, could I introduce Professor David Attwell.
Thanks, Jeff. Can you hear me at the back with the microphone? So, Ken Myer made exceptional contributions to Australian society, both in the arts and the sciences. He helped to fund the Florey Institute, which is promoting this lecture. He served the government as an economist, and he promoted funding of the arts by the state of Victoria. He even modernised the National Library of Australia. And his philanthropy lives on in the form of the Myer Foundation. So he was a truly great Australian, and it's an honour to be chosen to give this lecture in his name. Now, if we could have the lights down, please. The title of my lecture today is Brain Power, and I'm going to be using this phrase in two separate ways, but as we'll see, they're related. I'm going to use brain power to mean our brain power, how incredibly clever we are, how we can do cognitive tasks really quickly. But I'll also use brain power to reflect the energy supply to the brain, its power source. Because as we'll see, the brain's brain power depends critically on its power supply. So the brain is a bit like a computer, and you'll be aware that if you turn off the computer on your desk, if you remove the plug from the wall, then it stops working. And the brain is a bit similar. It needs a constant supply of energy in the form of glucose and oxygen, which, as we'll see, are provided in the blood. Otherwise, bad things happen to the brain. So let's have a look at a human brain. This is one here. You can see the folds of the cerebral cortex, the most recently evolved part of the brain. And when it's turned over, you'll be able to see the bottom of the brain here where the spinal cord would go off to control our arms and legs. Now, this few pounds of meat is what we perceive the outside world with when we look with our eyes or listen with our ears. We think with it. All of our thoughts are generated within this organ here. It controls our muscles to let us move through the world. We fall in love with it. It holds our deepest fears, but also our highest hopes. So this is what makes us truly human. And in the next 45 minutes, I want to tell you something about how this organ works, but also how it's powered. So the theme running through my lecture is that our brain power depends absolutely on the power supply to the brain. So I want to start out by showing you an example of the cognitive powers that a brain is capable of. We use our brains every day to do different things cognitively. If you want to cross the road in Melbourne, you're constantly assessing whether there are buses and trams coming, that kind of thing. And you need your brain to work quickly to be able to navigate that task safely. Now here's an extreme example of this from the Star Wars movies. For those of you who are either too old or too young to remember Star Wars, this is Anakin Skywalker, who turned out to be a Jedi Knight. And we'll see him in his Jedi Knight phase later. But here he is at the age of 6, carrying out a difficult cognitive task, slightly more complicated than crossing the road in Melbourne. And he's in a race, pod racing, against his arch enemy, Zabulba. Now, that wasn't Anakin dying, and we'll come back to Anakin later, because we'll see that Anakin's cognitive powers, the Jedi Knights, as some of you will be aware, had what was called the Force, and the Force was what allowed him to navigate this course so cleverly. We'll see later that the Force is related to the powering of the brain. So let me just give you a quick overview of what I'm going to talk about in my lecture. I'm going to start out with birth and end up with death with the aid of Superman, and go through a number of interesting themes in between. So I'll first of all look at how much energy brains use, and I'll then look at how they evolved to be able to use energy so efficiently to do information processing quickly. With the aid of Superman's monster, I'll try to convince you that the brain uses electrical signaling to provide us with our cognitive powers. And then I'll look more scientifically at how nerve cells use electrical voltage changes to encode information. I'll show you that this encoding of electrical signals in neurons is really important with the aid of these South American Indians and a really vicious snail. And I'll then come back to Anakin to explain where the energy comes from to power our very clever thoughts. And in fact, the energy is generated by these little creatures which live within us. Getting towards the end, I'll show you that we can look inside the living brain, image our thoughts, if you like, using the fact that the glucose and oxygen has to be increased to generate more energy from these little creatures. And at the end, in the tragic part of my talk, with the aid of Superman, I'll consider some diseases which occur to the brain when its power supply is cut off. So let's start out with birth. I want to introduce you to Tony. As you can see, Tony, who is the son of a PhD student of mine, is a voracious eater. And here he is knocking back his favorite tipple. And he cries a lot if he doesn't get this. And the reason he wants so much of it is to power his impressive cranium. So it turns out that 60% of Tony's food goes to power his brain. So kids' brains use a lot of energy. What about adults? Well, here's a typical adult with their brain fixated on the things that all of us are fixated on. And it turns out that for Homer, just like for us, his brain is about 2% of his body's mass. But his brain uses 20% of his resting energy. So as you're sitting here in the audience, 20% of the meals you've eaten today are being used to power your brain. Now, of course, Homer is resting all the time, so that means that 20% of his donut intake goes to power his brain. So the important thing to come out of this is that the brain uses a disproportionate amount of energy that the body produces. Okay, so thinking is very energy intensive. How much energy does the brain actually use? Well, it's about the same as a 20-watt light bulb. That's a rather dim light bulb of the old sort, or about what you get with one of these newfangled low-energy light bulbs. So that's how much energy is being used in your brain all the time. Let's compare that with another computational device, the computer on your desk. The computer on your desk uses about 10 times as much energy as your brain. Now, the computer on your desk is better at some tasks, a small number of tasks. In particular, if you want to search through long lists of things, like when you're doing a Google search to find a piece of information, then the computer's very good at that. But the computer thinks far less than our brains do. It's far less good at doing complicated tasks, like, for example, recognizing your granny no matter what angle you see her from. We find that extremely easy. Trying to program a computer to do that is much harder. So the energy use of the brain is high compared to other parts of the body. I want to look now at how our brain evolved to be able to use so much energy. And I want to look at brain evolution not over the time scale of the dinosaurs, which is of the order of 100 million years, but just over the last 4 million years as we evolved from the lower primates. Now, as we evolved from the lower primates, our bodies got bigger and our brains got bigger as well. So here's some brains from different species. This is a rat brain, a rhesus monkey brain, a chimp brain, and a human brain. And you can see as the bodies of these animals get bigger, so do their brains. The dolphin brain, interestingly, is even bigger, but I'm not going to talk about that today. But it turns out that you can extrapolate from the lower primates, extrapolate up to our body size, and try to predict how big our brain should be. And when you do that prediction, anthropologists have concluded that our brain is 3 times larger than you'd predict for our body size. And this is why we're so clever compared to the other species. So the larger brain is going to use 3 times more energy as well. You've got 3 times as many cells in there thinking powerfully. They're going to use 3 times more energy. So if the brain is going to use 3 times as much energy, then something must have evolved to use less energy, to be more efficient. And anthropologists have concluded, and I'll show you the data for this in a minute, that what evolved to use less energy is the gut. So here's their data. What these anthropologists did was to try to predict for humans how big the different organs should be by starting with the lower primates and extrapolating upwards for body size. And they predicted the brain ought to be about 400 grams in weight, whereas in fact our brain is about 2 kilos in weight. It's about 1,200 grams in weight, so it's 3 times bigger. For the gut, they predicted it should be about 2 kilos in weight, but in fact our gut only weighs just over 1 kilo. So the gut has got much smaller and more energy efficient. And we'll come back to why that is in a minute. The other organs all had about the size that they predicted. So these anthropologists suggest that what's happened is, over the last 4 million years, primate brains, ending up with ours, have got larger and use more energy by virtue of the fact that the gut has evolved to get more efficient and get smaller. So how is it that the gut can get more efficient? Well, it's thought that there are 2 contributions to this. I apologize to the vegetarians in the audience to start with. It turns out that part of this is thought to result from a higher nutrition diet with less leaves and more meat, which presents at least protein to the gut in a more readily digestible form. And the other thing that may contribute to this is a cultural change, the discovery of fire and the introduction of cooking, which again presents food to the gut in a way which allows it to be digested with using far less energy. So overall then, the gut got smaller and more efficient, and that allowed the brain to use more energy, which let us have many more neurons, nerve cells, so we can think more cleverly. Now, what do we use this energy on? How does the brain actually think? I want to introduce this subject with the aid of Dr. Frankenstein's monster, and we'll come back to him in a minute, but the theme is I want to persuade you that the brain uses electricity to think, just like computers do, in fact. So let's just briefly review a few facts about electricity. You may remember that Benjamin Franklin, one of the founding fathers of America, found that if he flew a kite into a thunderstorm and the string of the kite got wet and he had a key at the bottom of the kite, he could point it at the ground and electricity would arc from the end of the key to the ground. This is obviously a rather dangerous way of obtaining electricity, and he was lucky to survive, but he went on and did great things. Now, 30 years later, a much safer form of electricity had been invented, the battery, in Italy, and Galvani showed that if you took a battery and applied its voltage across the leg of a frog which had recently been killed, then when the voltage is applied to the frog's sciatic nerve, the leg twitches like this, okay? It twitches. So putting electricity into the frog animates it in some way. Not to be outdone, his nephew, 20 years later, applied this same technique to the recently beheaded bodies of criminals. And he found that if they'd been sufficiently recently beheaded, then again, applying electricity to their nerves would make their bodies twitch, and if he was lucky, they would rear up off the operating table in a satisfying manner. People would scream and faint, and he made a vast fortune exhibiting this to the general public. But the important scientific fact he discovered was that electricity animates humans. And this was carried to its logical conclusion with Mary Shelley's book, in which, and this is a clip from a still from the 1931 film with Boris Karloff. In this book, Dr. Frankenstein took a dead body, here's the dead body, with a dead brain transplanted into it, and with the aid of his faithful dwarf assistant, he raised this body into the path of an oncoming thunderstorm, and when it was up there, the brain was struck by a bolt of lightning, the body was brought back down, and as it started to move, in the movie you hear Dr. Frankenstein saying, "It's alive, it's alive." So again, electricity animates humans. So doubtless you've been convinced by these cultural suggestions, but I now want to just show you one more thing about this, which is that this idea of a spark of life animating people was actually a much older idea. When Michelangelo painted the roof of the Sistine Chapel in Rome, he depicted God here passing the spark of life into Adam. Now, we don't know whether Michelangelo was thinking about electricity, but we do know he was thinking about the brain, because it's been pointed out that if you look at this depiction of God here with all the putti around him, this is remarkably similar to the structure of the human brain. It's with God's legs being the spinal cord going off down here. This is for real. His shoulders here and this leg forming this band of fibers joining the two halves of the brain, the so-called corpus callosum. And the putti are the folds of the cerebral cortex, and even this apparently randomly placed scarf traces out the path of one of the blood vessels going to the brain. So what was he doing when he painted this ceiling? It may be that he thought, or he was trying to portray the fact that God needs to animate the brain of humans to bring them alive, or more subversively, it's been suggested he was playing a joke on his patron, the Pope. The Pope was extremely annoyed with Michelangelo because he was taking years to paint the bloody ceiling, 4 years, in fact. And so it's been suggested that he was trying to convey the fact that the idea of God is created in the brains of humans. Be that as it may, it's clear that over the centuries there has been a strong cultural suggestion that electricity is what animates humans. Now, I want to explain in rather more scientific detail how nerve cells use electricity to send information and to encode our thoughts. So here's a picture of some nerve cells. This is from a little bit at the back of the brain called the cerebellum, which controls our smooth movements. And here the nerve cells present have been labeled with different dyes so that you can see their structure. So if we look at nerve cells like this, we find they have two ends. They have an input end, which has this tree-like structure and receives lots of inputs from other nerve cells. And they then have an output end, the so-called axon, which sends out information to another nerve cell or to a muscle. So let's see how these cells encode information and pass it from one cell to another. I've just redrawn the nerve cell. This is its input end, and this is its output end. And if we zoom in on the membrane of the nerve cells are composed of a kind of plastic bag, this blue stuff, the membrane, which separates two compartments. There's salty water outside the cell, and there's a different kind of salty water, where the salt is based on potassium, not sodium, inside the cell. And if you were to put a little voltmeter inside the cell, you'd find there's a negative voltage there, about -1/10 of a volt normally, that kind of size. And when the nerve cell gets excited, this voltage goes positive, which I'm going to represent by this little star, and that positiveness opens up little holes in the membranes, which are made by proteins, which have got a charge on a gate like this, so it's pushed open, and that allows more sodium to come in. Na means sodium. That opens the next gate, more sodium comes in, and here's the next one, more sodium goes in. And so in this way, an electrical signal spreads along the axon. And if you plot out a graph of the waveform of these voltage changes, they're about 1/10 of a volt in amplitude, and each one lasts only about 1,000th of a second. Okay. Now, one of the themes of my lecture is the energy supply to do these kind of things, and it turns out that the energy is used on pumping out these sodium ions, which are mediating the electrical signaling in our brains. And about 1/4 of the energy being used in your brain goes on reversing this influx of sodium ions afterwards, so that you can do another one. Now, I want to show you an example of this electrical signaling, how it works in action when you see things in the outside world. So an electrical signal is going along the nerve cell, making these little spikes of voltage. Now, think about what happens when you're looking at something, as you're looking at the screen now. If you look at something in the outside world, then the eye converts that into an electrical signal, like I've just shown you, and it eventually ends up at the back of your head, the part of the cortex which deals with vision. And I'm going to show you a classical recording of this, in which, in order that we know when the electrical signals are happening, the electrical spikes have been converted to a sound. And this is a movie from two people, Hubel and Wiesel, who got the Nobel Prize for showing how the visual cortex analyzes information coming in from the outside world. Can you hear that? So here they're moving a spot of light across a screen and recording from a neuron, a nerve cell at the back of the brain, and then mapping out exactly where it responds. So it responds between these lines. You can see it likes it to go in one direction. This was recorded from the brain of an anesthetized cat, so you might imagine this cell is trying to detect little white mice moving in that direction. And Hubel and Wiesel showed that the visual cortex is designed to look for little spots of movement like this, but also for bars of light moving and for corners of light. And by having cells which respond to these increasingly complicated stimuli, you build up a picture of the outside world in each little part of the visual world, which lets you know what's going on in the visual world. You can see this cell doesn't respond so well to bars. It likes spots much better. So this is the cellular basis of perception, if you like. It's these electrical signals within the nerve cells which allow us to see the outside world. So I've told you about what happens with a signal going along one nerve cell, but when it gets to the end of the nerve cell, what's going to happen then? Well, it turns out that there are gaps between nerve cells called synapses, and the information goes across the gap by virtue of the nerve cell releasing what's called a neurotransmitter, which typically in the brain is glutamate. That's monosodium glutamate, like you eat in Chinese food or use as meat tenderizer. So this stuff gets puffed out, and it then excites the next cell. Now, how does it do that? If we look in detail at this gap between the cells, when an impulse arrives along here, it releases the glutamate, which binds to a little protein, the so-called glutamate receptor, and opens a hole that positive charge goes into and excites the next cell. And this is how information goes from one nerve cell to another, and we'll see why it's important in a minute. Then all these positive charges have to be pumped back out again, and it turns out that these use about 50% of your brain energy. So the great majority of energy used on signaling in the brain goes on these synapses. Now, what's the use of this? Well, it turns out that most of our thinking, if you like, occurs at these gaps between the synapses. And here's a nerve cell which is receiving input from two other neurons. Now, let's suppose you're hungry. You didn't eat before you came to the lecture. So there's a nerve cell somewhere in your brain saying, I'm really hungry. Okay, and it's sending information down here, but maybe the amount of glutamate released at that synapse is not enough to excite this other neuron. Similarly, There may be someone in the audience who isn't hungry, but who's got a bar of chocolate in their pocket, and they've got a neuron that's got that information encoded, and it's being sent down the nerve cell, but it doesn't release enough glutamate to excite the next cell. But for the person in the audience who's hungry and has chocolate in their pocket, they're in luck, because the information going down both of these nerve cells may produce enough release of glutamate to excite the next cell and produce a novel behavior. You remove the chocolate from your pocket and eat it. Now, this is a trivial example, obviously, because there are only 2 nerve cells involved. But typically, the nerve cells in your brain receive about 30,000 inputs from other nerve cells, and they add and subtract them, and on the basis of the signal that's produced, they decide whether to start a new behavior or not. So you can see from the number of synapses impinging on each nerve cell, there's a lot of scope for complicated thoughts going on there. And so from what I've told you so far, most brain energy, about 3/4 of it, is used on pumping out the ions that mediate signaling going along the nerve cells, that's 25%, and between nerve cells, that's about 50%, like that. So this signaling underlies all of our thoughts. So I want to give you an example of this for movement. I've shown you how you perceive things with the movie of the visual cortex. I want to turn now to how you initiate a movement. Now here's a movement. I couldn't resist showing this. This happens so rarely that it had to be mentioned. But this is, this is the last Australian batsman being bowled out in the last Ashes. Now, when a movement like this is initiated, it starts off in the top of your head in the motor cortex. And so what happens is a nerve cell up there gets excited, and it sends information down your spine, where it goes across a gap in the way I've just told you, and excites another nerve cell, which goes out, for example, along your arm to where you're holding the bat. And another neurotransmitter is released and makes the muscle contract to try to hit the ball with the bat. So do these ion movements matter? I've been going on and on about electrical movements in the brain, and I've been trying to persuade you that this is how you perceive things and how you move. I want to try to convince you with a couple of examples now that these movements really matter. So I'm going to start out by looking at these South American Indians. So these guys have a problem. They live in the forest, and most of the animals they want to eat to get a high protein intake live up in the trees, and they can't reach them. So to deal with this problem, they've developed these high-power blowpipes, which they project little darts out of with poison on the end of them. And the poison, curare, which is on the end of the dart, disrupts electrical signaling between the nerve and the muscle in the animals that they're trying to kill. So it blocks the signaling, the spread of the electrical signal from the nerve to the muscle cell. And what that's going to do is, first of all, make the animal lose its posture, possibly, and fall out of the tree. But even if that doesn't work, it will stop the animal breathing, because you need your muscles to breathe. And so after a few minutes, they'll die and fall out of the tree. And then they fall to the ground. You can pick them up and eat them. And luckily, the poison which is put on the arrows doesn't get absorbed across the human gut, so you can eat the poisoned animal without dying yourself. So the electrical signaling is clearly very important to those animals being killed, because if you disrupt it, then they die. Now I want to give you another example of this. This is a cone snail, and I'm going to call him Conan, because he's a barbarian. And Conan also uses a toxin which disrupts electrical signaling. And Conan is big friends with a fish called Nemo. Now, don't get too attached to Nemo. Nemo is a bit stupid and allows Conan to put his poison-tipped proboscis into him, and this disrupts the electrical signaling going on in Nemo's body, and unfortunately it's goodbye Nemo, and it's breakfast for Conan. This is speeded up twofold. Okay, so a bit of indigestion at the end there, not surprisingly. So this electrical signaling matters. So what I've shown you so far is that brains, especially babies' brains, use a disproportionate fraction of the energy generated in the body. They evolved to do that by virtue of the gut becoming more efficient, And most of that energy goes on electrical signaling, as Frankenstein showed us. And this signaling is used for perception and for controlling our movements, in fact, all of our thoughts. Now, I want to look in some detail now at how energy is generated within the brain in order to power this electrical signaling, because we'll see at the end, when I start to deal with disease, that it's a failure of the energy supply which creates a lot of the diseases which afflict especially our increasingly aging population. And this is Anakin Skywalker again, the guy you saw doing the pod racing, but he's now grown up into his adult morphology. We're going to come back to him in just 1 second, and he's going to explain where brain energy comes from. But first of all, if you want to improve the energetics of your brain, if you want to make your brain more powerful, where better to look than on the internet? So it turns out there's lots of stuff you can buy on the internet to give your brain vitality, to elevate it, to give it power, to fuel it, to give it energy or attention, or more subtly, to modulate it. If you look at the ingredients in these things, none of them are of any use whatsoever. But there is one substance you can go out and buy, probably in the foyer, which will give your brain energy, and it's Lucozade. They do sell that in Australia, don't they? Okay. So here it says, brain energy in a bottle. And if you can read this at the bottom, it says clear thinking in a bottle. And you can see it goes into the guy and lights up his brain. Now, can this possibly be true? Well, there was a court case about this in England because people asserted this could not possibly be true, but the court ruled, the Advertising Standards Authority found that this was a valid claim. And the reason it's a valid claim is that the main ingredient in Lucozade is glucose syrup, and as we'll see in a minute, glucose is the main fuel of the brain. So the fact that Lucozade is stuffed full of glucose meant that they were allowed to claim it was clear thinking in a bottle. Believe it or not. I should say that, of course, there's loads of glucose in your diet anyway, and you don't need to rush out and buy Lucozade. Now, where is brain energy derived from? How do we use this glucose in order to power the electrical signals which underlie all of our thoughts? I want to go to the myth of Anakin in the Star Wars movies. Here he was when he was pod racing, then he became a Jedi warrior, but tragically, he turned to the dark side and became Darth Vader. Now, his power is called the Force in the Star Wars movies, and very luckily, in the later prequels to the Star Wars movies, a biological explanation was provided of the Force, and I'm going to show it to you now. So you need to pay attention to how the Force is generated. Well, that would be bad. So what's this all about? So his power is attributed to this high concentration of midichlorians living symbiotically in his body and giving him energy. So what are midichlorians? Well, this is a midichlorian, and you're stuffed full of them. So in the Star Wars nomenclature, midichlorians are a kind of hybrid of mitochondria and chloroplasts. Mitochondria are the power plants of animal cells, and chloroplasts are the power plants of vegetation. So for mitochondria, it's believed that they're like bacteria, primitive bacteria, which were imported into our cells about 2 billion years ago, and they live symbiotically there in the host cell, and they're still here today. And the deal which they struck with the host cell was that the host cell would give them nutrients, and in return, they would make energy. Okay, so this is where brain energy comes from. This is what powers your electrical signaling and your thoughts. So how does it work? Well, here's a rather schematic nerve cell shown in green, and here's the blood supply. All the energy to the nerve cells comes along in the blood in the form of glucose, like in the Lucozade, and oxygen. Now, in primitive animals, before the midichlorians arrived, the glucose was processed by chemical reactions, and it was used to generate a small amount of energy. But with the import of the midichlorians into our cells, that allowed the products of those reactions to be combined with oxygen to generate lots of energy. So about 90% of the energy generated in our cells is made by these midichlorians. And this is the energy which is used to pump out the ions which underlie the electrical signaling in our brain, underlie our thoughts. And we can recognize that this oxygen-dependent mitochondrial pathway for energy production is very important because the brain has very little energy stores, and you know that if you cut off the oxygen supply to the brain, if you try diving deep in the swimming pool for too long, then your brain doesn't survive unless you go back up to the surface. So it's the midichlorians that give you your brain power. So here's a mitochondrion. Now, how do you decide where to put the mitochondria in the nerve cells? You know that in your house, if you want to run a refrigerator in your kitchen, you need to run a power lead to the kitchen and then to the refrigerator. If you want to run a TV in the living room, you have to have a power supply there as well. And it's the same in cells. They use energy in different places. And I told you earlier that much of the signaling energy is used at these gaps between the synapses, the gaps between the nerve cells, the so-called synapses. So how do you decide where to put mitochondria in cells? You need a mechanism to target them to where the energy is being used. Now, this is a mitochondrion walking around a cell. And they walk around on these microtubule rails pulled by a motor which really walks along like this. And the motor tugs on the mitochondrion by virtue of an adapter protein, which glues the motor onto the mitochondrion. So here are some real midichlorians moving around a nerve cell. You can see each of these white dots is a mitochondrion. It's been labeled with a fluorescent dye. And you can see they run around quite quickly, although the movie is speeded up 30 times. So some of them move around a lot, others move a little bit, and some of them are just parked. The ones that are parked are in parts of the nerve cell where lots of energy is needed. So how do you arrange this to happen? What targets them to the sites of energy need? Well, here's a nerve cell again with its input end and its output end. Here are the synapses I told you about earlier, where glutamate is released and generates an ion influx into the nerve cells. This is where half your brain energy is used, so you want to park the mitochondria near these synapses. And as I've just shown you, the mitochondria run around all the time on these tracks, and the red motor at the bottom pulls the mitochondrion because it's glued to it by a green adapter protein. So how can you make this be parked at the synapse? Well, it turns out the activity of the synapse itself is what parks it there. Because one of the ions which comes in at synapses is calcium. And it turns out the calcium binds to this adapter protein and unbinds it from the motor, so the mitochondrion can drift over to the synapse and make lots of energy. So the mitochondrion gets parked at the synapse in order that it can provide energy there. And that frees up the motor to go and be used for something else. So brains use lots of energy. It's used on electrical signaling, and that energy is generated by mitochondria. But as I've shown you, the brain uses lots and lots of energy, just 10 times more weight for weight than other parts of the body. So you need to focus the energy supply you've got to the brain on the neurons which are most active. And that means you need to increase the local blood supply in parts of the brain where the neurons are very active. And it turns out we can use that in order to look inside the living brain and image our thoughts, if you like. I want to explain how we can do that and give you some examples of imaging inside the brain. So here's the blood supply to the brain. This is how the energy comes into your brain. Although it's electrical signaling, it's a plumbing system which brings the energy in the form of glucose and oxygen. And if you have a bit of the brain like this is a wiring diagram of the cerebellum, so-called, at the back of your brain, which controls movement. These nerve cells are being powered by glucose and oxygen coming in the blood, and the oxygen comes in joined onto hemoglobin, a protein which you've probably heard of because if your hemoglobin goes low, you've got anemia. And the oxygen then comes off, leaving deoxygenated hemoglobin. Now, when the neurons are more active, they need more oxygen, so they send a signal over to the blood vessel saying, hey, dilate, bring in more oxygen and glucose, please. And this signal is mediated by the neurotransmitter glutamate, releasing certain messengers, either from the nerve cells themselves or from auxiliary helper cells called glial cells. So then the blood flow increases and more oxygen comes in, and you can think more with that little bit of your brain. So here's a movie of blood vessels being controlled by active neurons. So you can see the walls of the blood vessel here, and there are red blood cells moving around inside. These are what are bringing in the oxygen. And the green flashes are increases of calcium concentration in these helper glial cells. And they're releasing, when the neurons are active, these flash more and they release messengers onto the blood vessel. And so if the nerve cells want more energy, they dilate the vessel. And if they want less energy, they constrict the vessel. So it's by adjusting the diameter of the blood vessels in your brain. This is going on all the time as you're looking at the screen now. There are vessels changing their diameter like this to adjust the amount of energy supply to the different parts of your brain, depending on which nerve cells are active. And we can use this to make maps of the brain in which we image the parts of the brain which are active, shown as these yellow and red splodges. And the way we do this is to put someone's head into a big magnetic field in a scanner like this. And what this big magnetic field does is it allows us to study how the protons, the hydrogen atoms, the hydrogen nuclei in water molecules, H2O, absorb and emit radio energy. So here they are, the little hydrogen nuclei. They've all been lined up by this magnetic field. And then in this technique, you put in a radio signal at right angles to this other magnetic field, and that displaces the little nuclei a bit. And as they wobble back again, they give off a radio signal, which you can pick up. But it turns out that the radio signal they give out is affected by the amount of deoxygenated hemoglobin, which is nearby, because that's got iron in it, and iron acts like a little magnet, which affects the field they're in. And so when the nerve cells are active, that brings in lots more fresh blood, which lacks deoxygenated hemoglobin. And that alters the signal which comes out and lets you produce maps like this of your thoughts. So what thoughts are we looking at here? Well, these are the parts of the brain which are active when you're in love. Of course, there are two types of love. Not good love and bad love, but maternal love and romantic love. Now, romantic love is typified here by this classic picture from the Hôtel de Ville in Paris, showing French people doing what French people at least think they do best. And maternal love is symbolized by this Raphael picture from the Renaissance. And there's a couple of interesting things that come out of this. First of all, the parts of the brain which are active for maternal love, shown in yellow, and for romantic love, shown in red, are on the whole completely different. They do overlap in one little place and make orange, but mostly they're different. So that tells us that maternal love and romantic love, despite the same use of the word for it, are completely different things, conceived by different parts of the brain. And maybe that's because maternal love is much less selfish than romantic love. Secondly, if you just focus on romantic love and compare men and women when they're romantically in love, it turns out they use the same parts of their brain to be romantically in love. So although the man and the woman may be thinking very different things, at least they're using the same part of their brain to do it. Here's another example. If you see a scary face or something that makes you feel dread, then the part of your brain that lights up is called the amygdala. So in this way, by using the energy supply to the brain, you can image which parts of your brain are active. So I want to turn now to death and destruction and disease with the aid of Superman. We're going to come back to Superman shortly, but I want to start out by talking about stroke. This image on the left is a blood flow diagram of a human brain of someone who's unfortunately having a stroke in the scanner. And the red hot colors show where there's lots of blood flow, and the blue is where there's little blood flow. And here you can see there's a big area where there's no blood flow at all. And that's because a clot has come round from somewhere else in this guy's body and blocked a blood vessel, and there's now no glucose or oxygen going to this part of the brain. And if you go back and scan this guy a day later, you find that this is a structural scan now, that in that area there's a vast area of dead nerve cells. So that bit of the brain isn't going to work anymore. And stroke is obviously bad news. It affects 1 in 450 people a year. And as I just explained, it can be caused by block of a blood vessel, or in a smaller fraction of cases, about 15% of cases, blood vessels can burst if your blood pressure gets too high or the vessel gets too weak. So there are also other diseases caused by a loss of brain energy supply. For example, if you're riding your motorbike and you fly off and hit a lamppost and you damage your spinal cord, one of the main causes of damage is that you crush all the blood vessels in your spinal cord and lose the blood supply. And for children who unfortunately develop cerebral palsy, one cause of that is that when they're in the mum's womb beforehand, if the placenta isn't providing an adequate supply of energy to the developing baby's brain, then they can also have a problem. Now, this is the statistics for stroke incidence in Melbourne, taken from the kind of work that Jeff Donnan, who introduced me, does. And what it shows on the left is the number of people who have a stroke out of every 100,000 people in Melbourne. How many people have a stroke each year? So if you're young, things are looking good. But when you get up to be about 80, then you can see about 1,500 people each year out of 100,000 will have a stroke. So over this 10-year period, 75 to 85, that means that about 15,000, about 1 in 6 people are going to have a stroke. And it's about 1 in 3 when you get to be older than 85. And the lifetime incidence of stroke in Melbourne is about 20% for women and 16% for men. And it's less for men because they tend to do crazy things and die of other things earlier. So it's because they don't reach so well this old age part of the curve where they're more likely to have a stroke. So stroke, I've told you what it's caused by. Let's see what it does. Well, typically a stroke leads to the loss of about 1.2 billion nerve cells, about 8,000 billion synapses, and 4,500 miles of nerve cell axon in your brain. Each of your brains has actually got about 100,000 miles of axon in it, so there's a lot of cabling in there. It's the third leading cause of death, and it leads to paralysis and failure to speak, of course. So how does it happen? Well, it's all to do with electrical signaling. So if you remember, nerve cells send out impulses which have to jump the gap between the synapse here by releasing a transmitter called glutamate. But after you've puffed out this signaling molecule, you need to terminate its action again. Otherwise, you'd go on sending the same signal to the next nerve cell. So you have to stop signaling after you've sent it out. And it turns out that's done by helper cells called glial cells, which have got a little vacuum cleaner in them, a hoover, which is constantly hoovering up the glutamate, which is being released by the nerve cells. And although it happens rather slowly in this animation, in your head, this little glutamate hoover removes all the glutamate in about 1,000th of a second after it's been released by the nerve cell. But just like the vacuum cleaner in your house, this vacuum cleaner needs energy all the time. Otherwise, if you turn off the energy supply, it slows down and eventually stops. And worse than that, it actually runs backwards and pumps out all the glutamate that it's previously accumulated. Okay, so this glutamate accumulates now between the nerve cells, and it's going to excite this cell here, even though there's no information coming down the first nerve cell. And what's more, it's going to be exciting it not just for 1 millisecond like it was doing before, it's going to be doing it for a period of tens of minutes. So if we focus in on what's happening to that nerve cell, there's loads of glutamate around, even though there shouldn't be because there's no signals coming. It opens these receptors and lets sodium and calcium into the cell, just like it would do normally, but the excessive amount of calcium which comes in kills the nerve cells. So this is why when you have a stroke, you become mentally or physically impaired. So maintaining the power to the glutamate hoover is crucial to stopping nerve cells being killed in the grey matter of the brain, the bit where the nerve cell bodies are. Okay, say you have a stroke. Why can't we do a brain transplant to fix it? After all, we can transplant everything else. If you want to do a heart transplant, you can take a heart out of someone in New York and fly it across the Atlantic in a bucket of salty water, cold salty water, and plug it into someone in Europe, and it'll work just fine most of the time. But if you try to do the same thing for brains, it doesn't work. Now, there are several reasons for this. The first is an ethical issue, and that is that there might be a confusion of who you really are after you've transplanted the brains, as shown in the famous duck-to-professor transplant. But the second reason why this doesn't work is that as soon as you take the brain out of the donor, you've lost its energy supply, lots of glutamate is being released, and the nerve cells will be dead even before you've got the brain to the airport in the taxi. I want to turn now to a different kind of disease, illustrated by Superman. Superman, of course, was a great guy to deal with emergencies. And in this clip I'm going to show you, he solves a load of problems by reversing time. But sadly, for the actor who plays Superman, things didn't work out so well because he fell off his horse, broke his neck, and was in a wheelchair for the rest of his life. So, broken neck is not a new problem. This is the oldest known description of a central nervous system disorder, and it's from the Egyptian literature of 4,500 years ago. And it basically says, if you come across someone who's broken their neck and they can't move their arms or legs, it's bad news, you can't treat them. And sadly, that remains the case today. And it turns out that damage in spinal cord injury often reflects damage to a substance called myelin, which I'll explain what it is in a minute. It's a layer of fatty stuff around nerve cell axons. And this is also damaged when disability occurs in multiple sclerosis, where you have repeated cycles of damage to the myelin, and in cerebral palsy, which I told you about earlier, if you don't have a good energy supply to the developing fetus, then the baby's brain doesn't develop properly, and it doesn't develop this myelin stuff. So what is myelin? Well, here's a cross-section of the brain. I've told you about the gray matter of the brain, where the nerve cell bodies are. That's these grayer bits in this image. But half the human brain is white matter. And what the white matter does is it makes up the information superhighways of the brain, whereby information is sent rapidly from one place to another. You've got memories stored on both sides of your head. And if you want to think quickly, when the tiger is about to pounce on you and eat you, you need to think quickly. It's no good if the tiger storage bit is over here and the runaway bit is over here. You need to associate them quickly. If you send information between the two halves of your brain using ordinary nerve fibers, it takes about 1/3 of a second to get from here to here. That's quite a long processing time. Whereas if you coat the axons, so-called, of the nerve cells with this myelin stuff, and this is done by cells called oligodendrocytes. That sounds complicated, but it just means a cell with a few branches. Then that speeds the impulse, so it only takes 5 milliseconds, 1/200th of a second, to get it from one side of your head to the other, and that dramatically increases our cognitive powers. So here's myelin being made. One of these oligodendrocytes is putting out this fatty sheath, which it wraps around the nerve cell, and this movie isn't working. Great. I'll just talk through what the other movie was supposed to show. That lets the impulse go incredibly quickly through the nerve cell process. And the way it does that is the myelin acts to decrease the amount of charge that you need to change the voltage of the nerve cell. So you don't need so much ions to come in to produce a signal, and therefore it goes quicker. But if you lose the myelin, as is shown here, then the impulse goes much slower and dies out altogether. And so you become mentally or physically impaired. And this occurs in diseases like multiple sclerosis, cerebral palsy, as I've mentioned, stroke, and also spinal cord injury. And so what happens in the white matter, if, like Superman, you rupture all the blood vessels in your brain, in your spinal cord, that leads to a loss of the energy supply to the white matter of the nervous system, and there are glutamate hoovers there as well, which run backwards and release glutamate. And the glutamate opens little holes in the myelin sheath, and that destroys the cells making the myelin. And that's why people with these disorders, stroke, MS, spinal cord injury, why they can't walk anymore. So both in the grey matter, in the nerve cell bodies and the dendritic tree I showed you, and in the white matter, you need constant energy supply to the glutamate hoover to prevent the cell damage which causes mental or physical handicap. Now, of course, these diseases cause lots of anguish, and just to give you an illustration of this, I want to read out an email which I was sent. It says, "Dr. Attwell, you've been recommended to me as someone who might help me. I'm 57-year-old male, left paralyzed after a stroke 22 years ago. Can you or your colleagues help lessen my paralysis? I can travel to you and pay for treatment." This is Paul from South Carolina. Any response or direction appreciated. But tragically, there is no cure for the majority of these diseases at present. And so the bottom line is that if we want to treat these diseases, which are going to become more and more important as the population ages, then we need to do more animal research of the sort that's done in the Florey Institute, for example, to address these problems of the human brain that are caused by the brain losing its power supply. So I want to sum up what I've told you today. Brains use lots of energy. They evolved to be able to do this by virtue of the fact that the gut became more efficient. They use all of that energy on electrical signaling, as Frankenstein's monster showed us, and voltage changes moving along these cells are what we think with. So all of our thoughts are generated by this. The Indians and the snail showed you the importance of this electrical signaling, and Anakin Skywalker showed you that the energy to power it comes from these mitochondria or midichlorians, which came into our cells 2 billion years ago. By looking at the energy supply to these mitochondria, you can image our thoughts in the living brain. And of course, if you cut off the energy supply to the brain, as Superman showed, very bad things happen. Thank you for listening to me, and may your brain power stay with you as long as possible.
David, thank you. I'm sure you'll agree with me that was just a tremendous lecture, and clearly we should learn more from movies than we do at the moment. We need to focus and learn as we watch. Now, as I mentioned earlier, we're very fortunate tonight in having the nephew of Kenneth Myer, Carrillo Gantner, here with us tonight, who's going to say a few words on behalf of the Myer family, and in particular give the Kenneth Myer Medal. As you may know, Carrillo is well known already and has been a significant figure in the cultural fabric of our community, having made major contributions in both the arts and philanthropy. Ladies and gentlemen, Carrillo Gantner.
Professor Donan, distinguished guests, ladies and gentlemen. On behalf of my cousin Martin Myer and his fellow trustees of the Kenneth Myer Bequest, it's my happy responsibility to thank Professor Attwell for his delivery of the 15th Kenneth Myer Lecture, Brain Power. As you just heard, I'm Carrillo Gantner and a very proud nephew of Ken Myer. I share Ken's passion for the arts and for our engagement with Asia, but somehow I missed out on the gene for scientific inquiry. Two of my sons have the gene, and Martin has this gene, but he's otherwise detained stimulating more brain cells, snorkeling with humpback whales in Tonga. That's brain power for you. Plagiarism in academic research and writing is a serious offence, so let me confess up front that when Martin asked me to fill in for him tonight, I agreed on the basis that he would assist me by guiding me in what to say. So I'm happy to report that he's been as good as his word, which means in essence that I'm only as good as his words. The purpose of the Kenneth Myer Lecture is to attract to Australia some of the world's outstanding academics and to give the Florey Neuroscience Institute's community and the general public an exposure to a level of intellectual debate and science that is not always available in Australia. With this wonderful lecture today, Professor Attwell has joined an esteemed alumni of Kenneth Myer Lecturers, including, amongst others, James Watson, Eric Kandel, Sherwood Rowland, Colin Blakemore, Susan Greenfield, and Peter Agre. Given the focus of the Florey, it's not surprising that most of the lecturers, including Professor Attwell, have been neuroscientists, and all have been truly outstanding leaders in their specific field of research. Excuse me. As you've heard, Professor Attwell's work is on the signaling between the neurons and glial cells, how the brain's energy supply is controlled, and in turn, how that determines the computational power of the brain. I have to confess I had to use Wikipedia to check what a glial cell is, which of course you all know are those cells that support and protect the neurons in the brain. In engineering terms that Martin would be familiar with, Professor Attwell's work is focused on the utility infrastructure of the brain, the understanding of which has important implications in areas such as stroke, cerebral palsy, and spinal cord injury, as we've just seen. The Florey Neuroscience Institute is undertaking outstanding research into brain function. As a couple of examples, Professor David Howells is investigating how cultured brain cells respond to the stresses of a lack of oxygen and glucose. This enables him to understand how the brain may be protected from a sudden loss of blood supply and develop new drugs for treatment of patients with stroke. Professor Stephen Petru studies the energy-dependent electrical behavior of cells when their surface structure is altered by the introduction of different ion channels, thus gaining a better understanding of the mechanisms of specific forms of epilepsy. The Florey is currently entering a new and exciting phase of its development. The co-location of the Florey Neuroscience Institutes, or the FNI, with the Mental Health Research Institute and with the University of Melbourne's team of neuroscientists will create the 5th largest centre of neuroscience research in the world, with over 800 scientists working on dozens of research themes, including epilepsy, Parkinson's disease, stroke, multiple sclerosis, and addiction. The FNI has 2 new research buildings, one on the Austin Hospital campus in Heidelberg and the other on the University of Melbourne's Parkville campus, are now complete. The Austin building was officially opened this past June, and the Parkville building is due to be opened shortly in this month. The completion of these 2 buildings and the coming together of 4 neuroscience institutes plus the University of Melbourne marked the culmination of 7 years of hard work by all concerned, and Martin wanted me specifically to pass on his thanks to the many people involved. He looks forward to seeing you all at the opening in 2 weeks' time. Over the past couple of years, the FNI has appointed a number of exciting new scientific teams to fill the new spaces, including those dedicated to the study of stem cells in collaboration with the Universities of Melbourne, Monash, Queensland, and the Walter and Eliza Hall Institute to establish the Australian Stem Cell Centre. The Florey has also attracted high-field MRI imaging experts in collaboration with the University of Melbourne to take advantage of the world-class facilities being developed at the Parkville and Austin campuses. Talented young scientists to lead research in understanding of emotions and addiction have also been recruited to expand the capacity in areas where outstanding research is already being undertaken. Of course, all this new activity costs money, and while I certainly wouldn't be so tasteless as to do a pitch here, Martin would. He's asked me to take this opportunity once again to advertise the fact that despite generous grants from Commonwealth and state governments and donations from private philanthropy, the FNI has not yet completed its fundraising mission for the new scientific teams and is still looking to raise multiple millions. So should anyone wish to consider assisting FNI or need to be persuaded about the benefits, you can contact Professor Jeff Donan, who'd only be too happy to meet with you. These developments at the Florey would certainly have pleased Ken Myer. He was passionate about medical research, although his point of view was that of a scientifically unsophisticated layperson. What kept his sparkling attention and enthusiasm for so many decades and led to his involvement in the Florey was the excitement of new discoveries and the belief that these would make a difference to the health and well-being of the wider community. It's a passion very much aligned with that of tonight's distinguished lecturer, Professor Attwell. Would you please join me in thanking Professor Attwell?
Thank you.
Hold on. It's now my happy duty to give the Kenneth Myer medallion, and I just hope I open it right way up. There we are. In commemoration of his delivery of the 15th Kenneth Myer Lecture.
Thank you. Thank you very much. It's heavy.
Ladies and gentlemen, thank you for coming. That concludes tonight's official proceedings. On behalf of Martin Myer and the trustees of the Kenneth Myer Bequest, I thank you all for coming here tonight and look forward to seeing you at future Kenneth Myer Lectures. Thank you.